Joining us next is Dr. Tanya Gilbert, who's Executive Director of Translational Research at Eikonizo Therapeutics. Tanya will be presenting on the development of a highly selective CNS penetrant small molecule HDAC6 inhibitor for treatment of ALS, and really cool PET imaging technology to measure target occupancy. Welcome, Tanya.
Thank you so much. And thank you to all the organizers for giving us this wonderful opportunity to present today. And we're also really grateful for all of the collaborations we have with several ALS ONE members. So Eikonizo is focused on developing small molecule therapeutics for neurodegeneration. Our lead program is focused around our development candidate, EKZ-102. EKZ-102 is a highly selective CNS penetrant HDAC6 inhibitor, and it offers a potentially disease-modifying approach for both sporadic and familial ALS. It works by correcting broad proteostasis defects and intracellular transport defects to preserve neuron function. And currently, EKZ-102 is in IND-enabling studies with clinical trials anticipated to begin late next year. So in ALS, intracellular transport within axons and broad proteostasis defects really contribute to neuron death and ultimately motor impairment. And the relevance of these pathways in ALS is well supported, as there are genetic mutations in the machinery of these pathways. There is also a wealth of evidence from human pathology as well as preclinical models. Our hypothesis is that if you correct axonal transport and proteostasis defects, this will benefit persons with ALS. HDAC6 represents a really promising therapeutic target to do this because it is involved in both axonal transport and proteostasis pathways through its catalytic domain 2 and ubiquitin binding domain, respectively. More specifically, HDAC6 deacetylates several cytoplasmic substrates that regulate intracellular transport, and it also promotes the autophagic clearance of ubiquitinated and misfolded proteins. In neurodegeneration, there's this toxic cascade where you have dysregulated intracellular transport, your proteostasis systems are overloaded, and in certain cases, there's also an imbalance of HDAC6 and resulting hypoacetylation. So HDAC6 can act as a lever to really restore homeostasis and dampen down that toxic cascade. When you inhibit HDAC6 with small molecules against catalytic domain 2, intracellular transport is stabilized, but the beneficial role of the ubiquitin binding domain is preserved. So HDAC6 can still clear those toxic protein aggregates. And there is preclinical evidence showing HDAC6 inhibition is neuroprotective. HDAC6 inhibitors have been in development for several years for various indications. However, first-generation compounds had several liabilities that precluded their use as therapeutics. These first-generation compounds, which were mainly hydroxyamic acids, they have poor PK and poor safety profiles. They weren't actually very selective for HDAC6, so they had a lot of off-target effects. They did not get into the brain. And for these collective reasons, they were really having poor predicted clinical efficacy. Eikonizo has developed a next-generation class of HDAC6 inhibitors to really solve these problems. We use the oxidiazole chemical scaffold, and this allows for high potency and the potential of once-daily oral dosing. Our compounds are very selective for HDAC6 relative to all of the other HDAC paralogs. So this lessens those off-target safety concerns and leads to a more broad anticipated therapeutic index. Our compounds get into the brain and the spinal cord very well, which enables their use for neurodegenerative disorders. And we have a clear plan for using multiple biomarkers to show both target engagement and modulation of disease activity to give us a better chance for clinical success. EKZ-102 is highly selective and CNS penetrant. On the left, I'm showing some biochemical inhibition data. Here we can see that EKZ-102 is over 1,000-fold selective for HDAC6 relative to the other family members. And this is in contrast to ricolinostat, which is a first-generation clinical candidate that was only around 7-fold selective, and this compound actually failed in development. On the right, I'm showing mouse pharmacokinetic data, and this demonstrates that EKZ-102 has high exposure in the brain that's well above our biochemical IC50 levels, and this supports, again, its application for neurodegeneration. We have tested our HDAC6 inhibitors in a collection of preclinical models that are relevant to ALS, FTD, and Parkinson's disease. However, we do recognize that unfortunately in ALS, there's not a gold standard model, and there's been a lot of problems in the field translating preclinical efficacy into the clinic. And so we have really tried to put together a very robust, comprehensive data package that looks at each of our mechanisms of interest using multiple models. So we combine the use of human iPSC-derived motor neuron systems that have endogenous mutations with primary cultures from rodents that might overexpress toxic proteins or be given toxic insults with select in vivo rodent models looking at mechanism. We have found that our compounds can improve intracellular transport within the axon. We can reduce the burden of a broad range of toxic aggregates, including SOD1 and TDP-43. We can increase neuron health and extend neuron survival. And we found that we can also decrease plasma NFL and improve motor function in vivo. So due to the time constraints, I won't be able to go through each of the bullet items here, but I am going to be showing you some snapshots of our data, starting with SOD1 model systems. So just as a reminder, in ALS, intracellular transport is dysregulated, and you're not getting proper transport of things like mitochondria or misfolded proteins. So in our initial SOD1 mouse study, we were extremely fortunate to collaborate with Dr. Bob Brown and Dr. Justin Lee. And we used some novel imaging technology from the Brown lab that combines a radio-labeled tetanus toxin fragment with SPECT. And this allows you to measure the net retrograde transport from the leg muscle up into the spinal cord. And we found that our compounds can increase bulk transport in vivo. We next looked at their impact on SOD1 aggregates. So we found that our compounds can reduce the burden of mutant SOD1 in an in vitro system. Next, we have an ongoing collaboration with Dr. Theo Hatzapetros from ALS TDI doing a biomarker study in the SOD1 mouse model. And so far, we found that our compounds can reduce the amount of plasma NFL, and we have additional readouts ongoing. And finally, we find that we can improve motor function as measured by performance-based tests in SOD1 mice. In addition to SOD1 models, we have also tested the impact of our compounds in TDP-43-related models. And for the work shown here, the studies using human cortical eye neurons were done in collaboration with Dr. Sami Barmada and Dr. Michael Beckier. We found that our compounds can increase the amount of moving mitochondria in axons. We then evaluated their effect on TDP-43 proteostasis. We find that they can reduce the burden of phosphorylated TDP-43 in the brain of a highly aggressive TDP-43 mouse model. In human iPSC-derived motor neurons with a TDP-43 mutation, we can reduce the amount of cytoplasmic or mislocalized TDP-43. And in these same motor neuron systems, we can also reduce the missplicing of TDP-43 gene targets, including STAFN2, which we think is very exciting. And finally, we are able to improve the survival of neurons that have TDP43 mutation about on par with isogenic controls. And so collectively, this preclinical efficacy data, along with the drug-like characteristics of EKZ-102, make us really excited for its advancement. However, to have a chance of therapeutic success, you really need to engage the target in the tissues that you care about. And so we have used positron emission tomography to demonstrate that we can engage HDAC6 in the brain. So on the left, I'm showing baseline blocking studies using our HDAC6 PET tracer tool, EKZ-002, in non-human primates. This tracer gets into the brain very well, as shown by the baseline image. However, if you pre-treat the animals with HDAC6 inhibitors, the signal is blocked. And that decrease in signal is indicative of engaging HDAC6 and showing HDAC6 target occupancy. Beyond PET studies, we can also measure HDAC6 target engagement in the blood using flow cytometry assays in PBMCs. So we can look at a direct substrate of HDAC6, which is acetylated tubulin. So here on the right, I'm showing some in vitro proof of concept data where our compounds increase the amount of acetylated tubulin, showing they engage HDAC6. However, importantly, they don't engage other HDACs, and they do not lead to an increase in acetylated histones, and that is showing that they're selective. So these flow cytometry methods are readily adaptable to the clinic, and we do plan to use them in our initial phase 1 studies to monitor target engagement and help us tune dose. We're fortunate that HDAC6 biology intersects with ALS biology in many ways. And so there are many relevant markers of disease activity across these pathways I have listed here that we can take advantage of. And I'm just showing one example for each, but our plan is really to have a robust suite of relevant biomarkers so we can get a read on how our compound is working in early clinical trials. So where EKZ-102 is today, it is going through IND-enabling development. And again, we do hope to be in the clinic towards the end of next year. Our first clinical studies will be phase 1 SAD and MAD in healthy volunteers. And then we will expand into a first in-patient study in persons with ALS to extend the data set, but also to really pilot those disease activity biomarkers. We're really excited for the promise of EKZ-102, and we hope that it can offer a promising therapeutic approach for both sporadic and familial ALS. And I'm very grateful to have had the opportunity to present this work to you today.
Thank you, Tanya. That was a really comprehensive presentation of so many different preclinical tests and assays. Really, thanks for sharing. I had one question that I wanted to address. You mentioned that you're seeing a rescue of the TDP43 missplicing events like Statman2. Can you speak to how you envision your HDAC6 inhibitor is getting to that result?
Yeah, that's a great question. So we hypothesize that our compound is really working to clear misfolded or aggregated TDP43 from the cytoplasm, which should free up normal TDP43 to go into the nucleus and to do its job. So we think we're kind of removing that toxic sink and getting that out of the way so that the remaining TDP43 can have its nuclear functions.
Okay, yeah, it's one of those. I actually had been interested in asking that question at an earlier presentation. I've heard different theories on whether or not TDP43 that is mature, that has left the nucleus, can be re-imported into the nucleus, whether or not it still has a nuclear localization signal peptide. So that's a really interesting question that I'm curious how it plays out. But thank you so much for sharing your data with us and giving us a great presentation.
Thank you.